So we have several classes of phylogeny, and what we want to know is how do each of these cover diversity and how do they increase our coverage of diversity and generally contribute to expanding coverage. So the classes, most of them are pretty obvious: morphological, single gene, multigene, combined, which is multigene or molecular and morphological, multigenome, or mix. The most problematic one was synthesis phylogenies. So now you could say, if you have molecular data, some of it's, say, newly produced, and some of it's a bunch of old stuff which has been already published, and this is synthesis. Now the problem is with that criterion, actually virtually all insect phylogenies use some old data, so everything would be a synthesis. So you'd have to have some kind of cut-off. So from the definition for synthesis, is based on my own experience trying to build them, and what seems to be the main challenge is the dealing with very different structural features of different data types or different subsets and different phylogenies. So, for example, you could have a very broadly sampled phylogeny, and say what's the relationship between different orders, and you could have a phylogeny just from a single family. So these are structurally very different data types, very different kind of information. So from my opinion, synthesis is how do you integrate these very different structures. So based on that criterion, I excluded very large multigene analysis supertree results of modest size. These don't address this main challenge, which is the structural variation, and generally modest sized phylogenies, which similarly can use a more traditional inference method. So synthesis specifically is integrating very structurally different results that we tend to observe in insect phylogenetics, which is very, very fragmented. So we need to next class all of the results into these. So here is the insect phylogeny database, freely available, and you can see the data type columns here: molecular, MG, multigene, omics, AD is adult morphological characters. So the column info tab has some more detail on these. So where it was more easily parsed, I would give more specific character, larval, morphological, and so on. Some of these codes I did change over time. I started off with DB for DNA barcodes and now use SG. Started off with phylo markers, PM, but that should be really multigene would be the main term for that. Here's the omics methods I have encountered. Maybe there's more I have missed. So this is the Perl script I used for parsing that list, and you can see here how I've combined these different things into the classes. So AHE, OM, RADSEC, transcriptome, UCE. I forgot which one that was, NGS and bus code. These all I gave the code O for omics, PMMG, G for multigene, MT mitogenome, morphological, all of these, for combined synthesis and barcodes. So single letter code OGM PCSB. So these go into a variable class and are stored in this hash. But printed into the output file, which is named NUIX as sample table. So you can see then each tree ID, again, tree IDs in the first column, tree ID and a single letter code for the class. And here's some counts for the classes here. This might not be the most up-to-date ones. So I move that into this file name NUIX sample table with a date after it. So we are reading into here this script that simple table we just wrote with the tree ID and the class, and storing here. So we are going through a list of all NUIX and checking the class here. Because in some analysis some are omitted. So again, going through the file list and pulling out this regular expression will pull out the year, and these are stored by year into this object. Then all the years encountered in these are pulled out of the tree IDs, and this is a numeric sort. So starting at the earlier years, you can see here, starting at 1975 as the first phylogeny I happened to database, and going subsequently through all the years. So for each year, pull out the tree IDs for that year, split them, and this gives a count. So in 2014, 42 publications for that year. Split that list and actually open the file. So here we're going through all the publications published in, say, 2016. Open them, and from the NUIX string pull out all the binomials, and we've already stored the class of each file. So we then, for each binomial, we're checking if that has been stored in any of the previous years. If not, then that's a novel binomial for the current tree. That's incremented here. And then we print to the output file the tree ID, the binomials, which data class, year, and the novel binomials for that tree. So that's the main job we're interested in for the moment. That's output into this file handle, which is details each NUIX. So you can see the tree ID Here's the class, mitogenome, the year published, and the number of novel. So 13 of the species in that tree were not present in any of the full compiled data from the previous years. That's hard. That's the simple definition I use for novel coverage. So here we're then reading this into R. Have this table in R. Oh yeah, on this plot, synthesis was way off the chart, so I didn't even bother plotting that on this one. But some simple processing. We can then plot a simple histogram for the novel counts for each class, and you can see the main the outlier being the mitogenome. I think we're familiar with this type of paper where they sequence a new mitogenome and just combine it with all the previous ones, and we accumulate all this body of phylogenetics with each with very similar taxon sets. Otherwise, multigene genomics not much different.